All right, today we're going to shrink way, way down to the nanoscale. We're talking about a technology that lets us see the world of the super, super small in a way that, well, it was basically science fiction until recently. We're going to see how scientists are creating these incredible 3D images of life itself without ever even touching it.
So let's just jump right in with the central challenge, right? Imagine trying to map the surface of a soap bubble with your finger. You can't do it. You'd pop it in a second. Well, that's almost exactly the problem biologists face. They want to see these amazing dynamic surfaces of living cells, but these things are unbelievably fragile.
And here, you can see exactly what scientists are up against. This isn't just a smooth surface. It's a bustling city of proteins and channels and all sorts of moving parts. The problem is, if you try to poke this with some sharp probe, you're going to destroy the very thing you want to study. For decades, this was a massive roadblock.
But what if you could map that soap bubble without ever making contact? That is the absolutely elegant solution we're about to dive into. It's a technique with a kinda complicated name, but the idea behind it is just beautifully simple. And that solution is called Scanning Ion Conductance Microscopy. Let's just call it SICM for short. Really, at its heart, it's a way to feel a surface from a distance. And because it can do that, it lets us create these breathtakingly detailed 3D maps of really soft things, like living cells, just hanging out in their natural watery environment. It is the answer to our soap bubble problem.
So how does it pull this off? Well, instead of a sharp, solid needle, SICM uses this hollow glass tube called a nanopipette. You can think of it like a super, super sensitive microscopic finger. And when I say microscopic, I mean it. The opening at the tip is often less than 100 nanometers wide. To give you some perspective, that's about 1,000 times thinner than a single human hair. It's just wild. Okay, so how does this magic finger actually feel the surface without touching it? The secret is a tiny bit of electricity. Now, the key idea here is actually pretty simple. The system sends a very small electrical current flowing through the tip of that pipette. That current is carried by ions, just charged particles, that are already floating around in the liquid. The whole trick to SICM is just measuring that flow of ions very, very carefully.
And this breaks it down perfectly. Okay, so in step 1, the pipette moves down toward the surface. Now here's the key moment. Step 2. As that tiny gap between the tip and the cell gets smaller, it starts to squeeze the flow of ions. It's like putting your thumb over the end of a garden hose, and that makes the electrical current drop. The moment that happens, a feedback system kicks in and pulls the pipette back up just enough to keep the current exactly the same. The computer records that tiny up and down movement, hops to the next spot, and does it all over again. You do that thousands of times, and boom, you've built a perfect 3D map of the surface. No touching, no damage. Just brilliant.
Now, that initial idea was absolutely revolutionary, but it had one big problem. See, if you want to watch life happen, you have to be fast. And the first version of SICK, well, it wasn't. So that started this whole new quest to make this amazing tool faster, sharper, and a whole lot smarter.
And when I say slow, I mean painfully slow. We're talking about measuring just a few pixels every second. At that rate, trying to get a picture of a single cell could take forever. You could forget about watching anything move or change, because by the time the scan was done, whatever was happening was long gone.
So the first big breakthrough was something called hopping mode. On the left, you can kind of see the old way, trying to just drag the tip across the surface, which was super slow and pretty risky. The new solution on the right was to have the pipette quickly dip down, take its measurement, and then hop up and over to the next spot. This little change in strategy was a total game changer.
And you might be wondering, okay, so how much of a difference did it make? Well, just look at these numbers. The early modes were chugging along at maybe 2 pixels a second. Hopping mode jumped that up to 60. But with modern tech, we're now looking at over 1,500 pixels per second. That's a speed increase of more than 750 times. It basically turns system from a static camera into a high-speed movie camera for the nanoscale.
But you know how it goes? You solve one problem and another one pops right up. Moving that fast created a new issue called overshoot. Basically, when the pipette is flying down really fast, there's a tiny lag before the system can tell it to pull back up. In that split second, the tip could keep going and bam, crash right into the cell. This, of course, led to even more clever engineering to predict and prevent this from ever happening. But here's where the story gets even cooler. Because once scientists nailed the whole speed and safety thing, they realized that same basic principle, measuring tiny changes in ion flow, could unlock a bunch of new superpowers for SICM. It could do way more than just map out a surface.
So yeah, the modern SICM is like a Swiss Army knife for nanoscientists. By analyzing that ion current in much more sophisticated ways, it can map a cell's squishiness, its mechanical stiffness, it can map its electrical charge, it can even map chemical activity happening at the surface. It's not just taking a picture anymore, it's doing a full diagnostic workup on the cell.
And this right here shows you that power perfectly. On the left, you've got the physical shape of the cell, the topography. But on the right, captured at the very same time, is a map of its electrical charge. Suddenly, you can see exactly how the structure of a cell is related to its electrical function, which is just incredible.
So how in the world does it measure stiffness? Well, it's really clever. As the pipette gets close to a soft surface, that flow of ions actually creates a tiny amount of pressure, enough to gently push on the cell membrane and deform it just a little bit. This changes the way the current responds as the tip gets closer. By analyzing that response, scientists can actually calculate how stiff or soft that exact spot on the cell is, pixel by pixel.
All right, so we have this fast, safe, incredibly versatile tool. What are people actually doing with it? Well, this is where the science just gets wild. Let's look at some of the truly groundbreaking stuff that SICM is making possible right now.
I mean, just look at this. We're not just seeing the surface of the cell here. By combining SICM with other techniques, we're actually peering through the surface to see the cytoskeleton. That's the internal scaffolding that gives a cell its shape and helps it move. And the amazing part is, this is all happening on a living, breathing cell.
And it's not just for biology. This tech is also a huge deal in material science. What you're looking at here is scientists watching the surface of a battery electrode as it charges and discharges. They can see it swell and change in real time, which is giving them incredible insights into how to build better, safer, and longer-lasting batteries for everything from our phones to electric cars.
Okay, this next one sounds like it's straight out of a movie. Scientists can now use the SICM pipette to perform what they call a nanobiopsy. They can guide that tiny tip to one specific spot on a living cell, apply a little bit of suction, and gently pull out molecules like RNA without killing the cell. They can then analyze that material to see what's happening genetically inside that one little part of that one cell. The potential for understanding diseases is just immense.
And here's another incredible combo. The SICM first acts like a scout, mapping the cell surface to find one specific ion channel, one of those tiny little pores that control all the electrical communication in our bodies. Then, a second tool comes in to listen to that single channel and record its electrical activity. This lets us draw a direct line between a cell's physical structure and its actual function.
Okay, so let's just boil this all down. If there are a few things to take away, it's these. First, SICM is this amazing, unique way to image delicate, living things without hurting them. Second, it's evolved like crazy, going from a slow, simple camera to a high-speed, multi-talented science platform. And finally, because of all that, it's giving us these revolutionary new insights in everything from medicine to new technologies.
And that really leaves us with one final, huge thought. For the first time, we have a ringside seat to watch life's tiniest machines as they actually work. We can see them in action, watch them respond, and even ask them questions. So the only thing left to wonder is, what amazing secrets are they going to reveal to us next? Thanks for tuning in.